So it's my own turn. I'll be discussing about pitfalls in the interpretation of clinical trial reports. So up until now, you have heard several principles. From this talk and onwards, you'll be learning about several actual tips on how to read a research, how to do research. You'll be learning about how to do peer review, what does an editor expect when you submit a journal, and how to write a good study, how to do a meta-analysis. So from this point onwards, all the talks will hopefully be much more relevant to you, irrespective of which discipline you are in. So I have no conflicts of interest. So let me start with this picture. This is the picture of our in-flight magazine for Nepali Airlines. When I came to Kathmandu from Mumbai, I took Nepali Airlines, and this was the in-flight magazine. I was very interested in reading what it has to say, and this is the last. This was one of the last pages of the magazine. And as you can see here, this is advertising several packages, health packages, basic health packages. If you can spend more money, you'll get a comprehensive package. Much more money, super health package, and premium health package. Under premium health package, irrespective of your age, you can get a BMD scan, you can get a PSA test, you can get all sort of these exotic tumor marker tests, TFT test. These are not for people with symptoms. This is package, like screening asymptomatic people, people with no problems, and finding that, wow, maybe your vitamin D is a little low. Oh my God, what's happening? Or maybe your PSA is a little high. Should we biopsy your prostate? Should we take it out and make you incontinent for your life? So these are all wonderful opportunities to overtreat, overdiagnose, and harm our people. And why did this happen? One of the reasons is we do not understand the clinical research and clinical trials behind these recommendations. So that's the reason why I think understanding how to read a clinical trial report well is of paramount importance. Because this is, honestly, this is all nonsense. We should be stopping this. Okay, so let me start with how do we read a clinical trial report. I'll use real life examples, but as an oncologist, I'll be using examples from cancer trials, but it will be applicable across. So let's say you see this report. This is a New England Journal publication. So first thing, simply because a paper is published in New England Journal or Lancet or JAMA does not mean you have to follow it. There will be several biases. Previous speakers have shown you how much bias creeps in this report. So just because it is in New England Journal does not mean it is a mantra. Does not mean you have to follow it. You can say no. Okay? So the first question is, when do you read the abstract? My answer is almost never. You should read the full paper. Sometimes, like, let's say there is a patient with metastatic pancreatic cancer, you want to use a chemotherapy called FOLFIRINOX, and the patient says, how long am I going to live? In that case, if you want to quickly go and see, then at that case, you can read the abstract. But don't read the abstract before you have read the full paper. Because this is a news that I read on Twitter. It says, Pakistan cricketer Mohammad Irfan confirms he's alive after death. What does that even mean? How can someone be alive after death? This is your abstract. The full paper says, confirms he's alive after death hoax circulates. Now that makes sense, because someone cannot be alive after death. So you have to read the full paper. Abstract is not enough. So a couple of things. Even before you start reading the paper, I think you should be aware of a couple of things. One is who funded the trial, who sponsored the trial. Are there conflicts of interest among the authors, and who wrote the paper? Which sounds weird, right? Who wrote the paper? The authors write the paper. But that's not always the case, as we'll see. Okay, real example, the same trial. So where do you find these details? This is the end of the abstract, in which it says it was funded by this company, Ross and Genentech. And then in the method section, you'll see that the trial sponsor, which is the company, Ross Genentech, it not only provides the drugs, which is normal, they can provide the drugs, but it also says they collaborated regarding trial design, which is also fine, data collection, fine, analysis, even up to that point I'm okay, but also interpretation. How can the industry interpret the trial it itself ran? That's like I am the judge of an essay competition where my own daughter is participating, and I say, my daughter is first. Of course, I'll say that she's first. She's my daughter. But the problem is, you should not make me the judge. Why did you make me the judge of an event where my own daughter is participating? Similarly, how can the industry itself interpret the data for its own drug? And then, in a different context, if you see there, it says, all the drafts of the manuscript were prepared by the authors with editorial assistance from professional medical writers funded by the sponsor. So the authors actually did not write the paper. The company paid professional writers to write the trial report. And yeah, so what do you think they will write? They will write wonderful things about their drug. So that's why, simply because something is in New England Journal of Medicine does not mean you can take it for granted. And then the other one is showing actually the affiliation of authors. So 2 of the authors actually belong to the company. So it's not just professional writers, but 2 of the authors actually are employees of the company. And depending on journal, you can find this information at different places. Like this is another example, olaparib trial. It was funded by the company, and you can also see the clinical trials number if you want to see details. But for conflicts of interest, you have to go towards the very end. It's weird. Sometimes the journals, they do not give conflict of interest with the abstract. So you have to actually pay for the full publication to see whether they are conflicted or not. So towards the end, you will see this. And as you can see, I have highlighted there. One of the authors was employed by the company. Someone has stocks in the company. So you should be mindful of all these things. Okay. Then trial conclusions. When do you read the trial conclusions? I think that you should make your own conclusion after reading the paper. I don't think you should read the conclusions of the authors, because people can interpret data in different ways. This is an actual news. A pair of glasses were left on the floor of a museum, and everyone thought it was art because it was in a museum. It was someone forgetting their glasses. So I'll show you an example. This is the conclusion of one of the trials, and you can look how much bias there is. Now you might or might not understand these nuances, but it says, although survival was in favor of our drug versus placebo among this group of patients, if they say although, then you should think that there is something fishy. They are going to cover it up. There was no statistically significant difference. So that means actually it was not difference, but they are saying there was actually benefit. And then they talk about a different endpoint, PFS2, trend. If you see this word trend to justify any conclusions, you should think that the data are not good. And what was not alpha protected. This is, we can discuss about this later, but this is an epitome of bias, of what a professional medical writer can do. If it was up to me, I would have written that this drug is not useful, we should not be using it. That would have been the conclusion. So be aware of experts. So not all experts actually give you right education. One of those experts, Kellyanne Conway, you may or may not know, during Trump era, she says, this is COVID-19, this is not COVID-1, not COVID-2, COVID-17, 18. What is the WHO doing? It started in 19. So we did a paper about, editorialists, experts who write editorials for these new trials in these big journals, because some people rely on the editorials to understand whether or not it's a good trial. But we found that the editorialists themselves have significant conflict of interest with the same industry, with the same company whose drug they are discussing. I find it unacceptable. And then you should also be mindful that some experts may be saying the right thing, but because of these conflicts, that voice can be drowned. For example, this is a joke, but if Galileo was alive today and he was using Twitter and he said the earth revolves around the sun, then the consensus was opposite at that time. So Twitter would say this claim is disputed by theology experts. So yeah, there is both pros and cons. So let's discuss a couple of issues to look out for when you are reading a clinical trial publication. The first thing is whether it's a randomized trial. So trials can be randomized trials or non-randomized single-arm trials where there is just one arm. Randomized trials is one of the best inventions of medicine, or not even medicine, it expands other disciplines as well. Because randomized trial, it's only the randomized trials that can give you the answer to the question, what would happen if not for this intervention? Someone says you have to use drug X in condition Y. What would have happened if we did not use that drug? That question can only be answered by randomized trials. You can't say, okay, we are using drug X in 20 patients and 15 of them responded. In the past, we know that only 5 of them would have responded, so this is a better drug. No, you cannot do that. You can make that conclusion only after a randomized trial. Single-arm trials are biased. They can use selected population. For example, in the past, that study might have been done in patients with severe liver disease, child poo C, and this one might have been done in child poo A. There might be age difference. There might be several differences related to ethnicity. There are so many variables that you can control, and there are so many that you cannot control. That can be answered only after randomization. So single-arm trials are not powered, and there is not even a good criteria for success. For example, let's say cancer drug. We use a drug in 70 patients. 20% responded. Is that good? Who knows? 25%. Is that good? Don't know. 18%? So there is no good criteria for success. And it's not meant to inform practice anyway. And in cancer, for example, we know that single-arm trials, they overestimate responses, response rates and duration of response compared to randomized trials. For example, this was a paper that we did in which we compared the duration of response for the same drug in the same cancer for single-arm trials and randomized trials. And we found that the single-arm trials showed 17% higher duration of response than the randomized trial for the same drug in the same condition. And this has been shown in the past with cytotoxic agents as well, with response rate. Phase 3 studies, they show smaller or lower response rates than single-arm trials. Then after that, after you have seen whether it's a randomized trial or not a randomized trial, the next question you'll ask is, are the patients in this trial, are they representative of my routine patient population? In other words, are your patients being represented? The patient that comes to your clinic tomorrow, would that patient have been a part of that trial if that trial was open at your place? And this is important because, again, the example is from cancer, but this is applicable elsewhere as well. For example, in cancer studies, we frequently exclude patients who have brain metastasis. We exclude patients who have HIV. We exclude patients based on age. We exclude patients based on the renal function test. We exclude patients based on the liver function test. So, but when the actual patient comes to your clinic, that patient has several problems. Bilirubin is high, creatinine is high, and you don't know whether that results will apply to this patient or not. Average number of eligibility criteria is actually 16 for cancer trials. And then again, about the representativeness, in this study, of which I was also a part, we looked at whether the trials that lead to FDA approval are representative of the US population, but we should think whether they are representative of our population. For example, female patients were underrepresented in these trials. Black patients were underrepresented. Obviously, Indian subcontinent patients are far underrepresented. And patients with HPV and HCV infections, they were only a few percentage of the patients. So we need to be mindful of the patient population. Why? Why should we be mindful of that? Why does it matter? Because if the patient population is highly selected, then it exaggerates the clinical benefit. I'll give you an example from hepatocellular cancer, liver cancer. If you look at the median overall survival here, for the control arm, not for the drug arm, forget about it, for the control arm, then you see survivals of 8 months, 12 months, 8 months, even in second line. While we know that the average survival, even in first line, is around 9 months. How are these patients in control arm who are not getting treatment, who are getting placebo, surviving for 8-9 months in second line? Because these are highly selected patients, what Dr. Booth referred to as the Olympian patients. In fact, if we take the trial of sorafenib in hepatocellular cancer, if you look at this curve and the data, then if you look closely, patients who got sorafenib lived for an average of median of 11 months, and patients who got placebo lived an average of 8 months in the trial. But when they looked at what's happening in the real world, they saw that the median survival for sorafenib was 5 months. That means patients getting placebo in the trial were living longer than patients who got the drug in the real world. Placebo was better in the trial than drug in the real world. Okay, we asked, is it randomized trial or not? We asked whether the patient population is representative or not. Next, we'll ask if it is a randomized trial, is the control arm appropriate? Because in randomized trial, you'll have an intervention arm and then a control arm, the experimental arm and the control arm. But the control arm should be the standard of care. If the control arm is not the standard of care, then you are testing your new agent against an outdated control. So how can a control arm not be appropriate? If we are using a control that is already shown to be inferior in previous randomized trials, or if we are not allowing an effective drug to be used in the control arm, or if we are using a placebo or ineffective drug instead of active or effective comparator. Some examples. Again, just for the sake of example, you don't have to understand oncology for this. For patients with lung cancer, there was a drug called pembrolizumab, which was approved in 2016. So this was a good drug. All patients who do not have problem with getting this drug would get this drug. But then they ran a new trial of a different drug called cemiplimab from 2017. That means at this time, pembrolizumab was already available. But the control arm for this cemiplimab trial was again chemotherapy. It was not pembrolizumab. So there was a better drug in the market, but patients were getting an old chemotherapy as the control arm treatment. That is inappropriate. And actually it got approval based on this, but we think that getting approval in the US based on using a trial that has a poor control arm that is being done in low and middle income countries is very inappropriate. And we have written about it more in this paper, which you can refer to. And the other example of inferior control is not allowing an effective drug. Now again, just for example, this was a trial of a new agent called olaparib in metastatic breast cancer. And here if you see, patients were randomized to receive this drug or standard therapy. They say standard therapy of physician's choice. So you might think that, okay, the physician can choose whatever control arm treatment they can give, but in bracket, they limit the option to capecitabine, eribulin, or vinorelbine. That means for these patients, there was actually another option called platinum, which would have been more effective, but by limiting the options, they are not allowing a better control. And then using a placebo control, because sometimes placebo is not appropriate if there is an active agent that does better. Again, a trial of pancreatic cancer. Here what they did was they randomized patients to receive chemotherapy after certain months to receive chemotherapy or placebo. In real world, for patients with metastatic pancreatic cancer, we don't give them placebo. We give them an active agent. So yeah, they used placebo, which was unethical, and no surprise, there was a difference in PFS, but no difference in overall survival. And then they said the quality of life was maintained. This is a fancy way of saying quality of life was not improved, right? So basically, it's a drug. Then the correct conclusion would be this is a drug that does not improve how patient feels or how long a patient lives, but costs more than $12,000 a month and has some important side effects. So is this a drug that's worth having? No. And then after looking at the control arm, the next question we should ask while reading a trial is, what is the primary endpoint of the trial? And you have heard a lot about this from Chris Booth's talk, so I don't need to go into detail. He talked about the use of surrogate. One reason why people want to use surrogate is because it shortens the trial duration. If you use survival, it takes a longer time. But that duration is not quite long as we would imagine. It's only a mean of 19 months if you use response rate, or 11 months if you use PFS. And the surrogate needs to be validated, as Chris showed, but in most cases it's not validated. The validation is poor, and as Chris showed, the validation with quality of life is also poor. And this is a recent paper that we did, and here I'm just putting this slide to say one thing. In cancer drugs, they make a big deal about oral drugs, and they say that, oh, the patient does not need to get injection, they are getting it orally, so this is chemo-free. And chemo-free treatment means wonderful thing for the patient. But actually when we look at the data, even if it was chemo-free, even if it was oral targeted drugs, it had important side effects and patients actually had worsening of quality of life. So chemo-free does not automatically mean better quality of life. So even in your discipline, if you have an IV agent and an oral agent, oral does not necessarily automatically mean that it's better than IV. So let's move on to the issue of statistical significance versus clinical meaning. We all know how important a p-value of less than 0.05 is. P-value of less than 0.05, you get a paper published. More than 0.05, you don't get a paper published. And many people actually do several maneuvers to get a p-value of less than 0.05. Sometimes they might think that maybe this patient was not important, let's exclude this patient, and then Eureka 0.049, and you can publish the paper. That's a wrong practice. We should not be doing that. And in fact, we should not even worship p-value of less than 0.05. It does not tell us anything about the magnitude of benefit. It simply tells us what's the likelihood of the results that we are seeing if everything, when controlling every other thing. What's the likelihood that these results are actually the effect of the intervention and not from chance. But in oncology, it has become a big problem. We are chasing just a p-value of less than 0.05. Based on this paper, I'll give you one example. It's the NALA trial in breast cancer. It's a trial of neratinib, a new drug, versus lapatinib. And here, these are the results. The median PFS was 5.6 versus 5.5 months. This is not a typo. The median PFS increased by using that drug by 0.1 month. That's like 3 days. If the patient missed one CT scan appointment and came back 1 week later, it would have been longer than this. So, but the p-value was less than 0.05. It was 0.0059, Eureka, Eureka. And it got FDA approved. And then, again, this is the issue about how medical writers can trickily write the results. The overall survival was not improved. But they write it as, although a numerical difference favoring this new drug was observed, statistical significance was not reached. This is just a way of making us fool. The correct conclusion here is, it does nothing for the patient. Overall survival does not improve. PFS improved by 0.1 month, which is just a fluke. And we should not be using this drug. It's very toxic for the patient. So one other thing I want to highlight here is, sometimes while we are practicing in countries like ours, we feel like, oh my God, we don't have access to this drug, that drug. But this also highlights that some of the new drugs are not even worth having. So we should not feel bad about ourselves that we are not having access to all these new drugs. Most of the drugs that are really good, we do have access to them. Okay, some other issues, some small issues when reading a clinical trial report. I'll not have time to go through all of this in detail. Like, there are issues about how to read a non-inferiority trial and what to do about trials that are stopped early. The issue of censoring, Chris mentioned in his talk briefly. But I'll take some time to discuss about subgroup analysis and publication bias. I think these are relevant across several disciplines of medicine. So publication bias, it can happen in several ways. One is obviously not publishing negative results. And that's how I'll talk about this in my talk tomorrow about how to do a systematic review as well. Because if you don't publish the negative studies, then the evidence base becomes skewed. But the other publication bias, one example of publication bias in oncology, for example, is about quality of life data. They give you the results of survival or PFS. They don't tell you the results of quality of life. So you keep wondering what happened to quality of life for the patients. And sometimes they don't publish at all. And sometimes they publish it after 5, 6, 7 years. And the main results they publish in New England Journal of Medicine. Quality of life data they will publish in a small conference abstract. So you can't even find it. So that leads to publication bias. In this paper that we did, we looked at 352 phase 3 randomized trials in oncology. And we found that 50% of them, they did not even bother including quality of life as an endpoint. Of those that did include, 25%, they did not report pre-specified quality of life endpoint. That means they said they were going to measure it. They measured it, but they did not publish it. So why do you think that happens? Because obviously the results were not good, probably. That's why they didn't want to publish it. So this study actually shows how long it takes for these results to be published. Even trials with positive results, only 15% of them have their quality of life results published within 1 year. Even trials with positive results, that means these are practice-changing trials. But even for those trials, we don't have quality of life data up until several years. And when you are reading an abstract, especially in the abstract, sometimes when they are summarizing the toxicity data, the side effect data, they will use certain terms to make us fool. So while describing side effects, they will say the side effects were acceptable. They will say the side effects were manageable, feasible, favorable, tolerable. The treatment is safe. They will use these terms. They will not give you the exact numbers, but they will use these terms to describe side effects. That's why we have to read the results, because these are meaningless words. These are just terms that are used to downplay toxicities. You know, if you read this paper, you'll see several examples, because we looked at these trials that were using these words, and we found that there were several patients who had fatal adverse events. That means patients died as a result of the side effect of the drug, and still they were saying the side effect was acceptable. Acceptable to whom? It was not acceptable for the patient's family, and they said the side effects were manageable. If you could have managed it, the patient would not have died. Okay, next, let's talk about subgroup analysis. This is an important concept, and I think this is very much prevalent in several disciplines of medicine. In cardiology, this is a big problem as well. So subgroup analysis is, they give you the results of the main, they give you the main results, but then sometimes when the main result is positive, sometimes even when the main result is negative, they look at different small subgroups of patients and they give the individual results for each subgroup, and then they will try to claim any treatment effect based on those subgroup analysis. One example, this is the affinity trial, adjuvant pertuzumab trial in breast cancer, HER2 positive early breast cancer. And this is what they found in the intention to treat, the overall population. Can you even see that there are two lines here? There is a red line and a blue line. That means this drug is so marginal, so useless, that you can't even appreciate that there are two lines. But it has a p-value of 0.045. It's less than 0.05. Eureka, eureka, got approved. But why would anyone want to use this drug? They would, because they presented subgroup analysis, ladies and gentlemen. Here you go. Out of several subgroup analysis they ran, one of those was based on nodal status. That means whether the lymph node was positive or not. And as you can see here, when the lymph node was negative, it was not significant. When the lymph node was positive, the result was significant. And again, here the difference is a little bit better than what it was. Here you can barely see that there are two lines. And so now they say, okay, you don't have to use it on everyone, but if it is node positive, use it. Is that the right conclusion? No, this is just a conjectural finding. This is just a guess. This is just a hypothesis. Because there were 12 exploratory subgroups. This is just one of 12 exploratory subgroups. There is a saying that if you beat your data enough, it will confess to anything. So if you run multiple analysis, one of them will turn out to be positive. This could have been such spurious findings. And the interaction p-value, actually they do give the interaction p-value, it's 0.17. The interaction p-value is not significant. When you are running such multiple analysis, it should be adjusted for multiplicity, but that was not done. And what happened here? Okay, so one more example, like these are just references so that you can study about this in detail if you want. This was a paper, a controversial paper we wrote in JAMA Oncology, because pemetrexed is a cancer drug that is recommended to be used in non-squamous, non-small cell lung cancer, but this was based on a subgroup analysis, and we call it a $1 billion subgroup analysis. And similarly, this is another example of how a subgroup finding, or how the statistical principles were violated to make a claim when it was not true. So I just want to give a recap of the points that I talked. So what should you look out for in a clinical trial report? You have a clinical trial report in front of you. You just printed it out from New England Journal of Medicine. Now you're about to read it. What should you see? First, conflicts of interest. Abstract, don't read it. Then look at who wrote the paper. then see whether it's a RCT or a single arm trial. Is it a phase 3 trial or even in RCT, sometimes it can be phase 3 RCT or phase 2 RCT. I did not go about that in my talk today. And then the randomization allocation. Usually it's 1:1. Sometimes it can be 2:1, 3:1. It's a little complex topic that we can discuss later. But more importantly, what's the eligibility criteria? Is the trial population representative of your patient population? Is the control arm appropriate? Is it the standard of care? Did the control arm get crossover? Again, crossover is another topic that we have to cover later. And post-protocol therapies. This is an important thing. After the trial was complete, what did the patients get after the trial was over? If there was a bias in post-protocol therapy, that can lead to confounding in the results of your primary analysis. And again, what was the primary endpoint? And was the results just simply statistically significant, or was it also clinically meaningful? So you can use this as a checklist of things to evaluate when reading a clinical trial report. But if you want to learn more about this, our Queen's Oncology also has a masterclass in critical appraisal. This is free. You can Google this. And there are lectures here from Dr. Chris Booth, Dr. Ian Tanak, and others, and you can read, you can go through these modules as well. Thank you. I'm happy to take questions.